Hey there. You know, every living thing has a kind of instruction manual, a biological blueprint that makes it totally unique. And for us humans, that blueprint is just incredibly complex. So today, we're going to dig into what happens when there's even a tiny little typo in that master copy.
Okay, it all comes down to this number right here, 46. That is the total number of chromosomes. Think of them like the individual instruction manuals, packed into almost every single cell in your body. It's the whole shebang, everything needed to build and operate, well, you.
So getting that copy right is absolutely crucial. Which leads to the big question, right? What happens when the cellular copying machine messes up? When there's a typo in this fundamental blueprint? That's exactly what we're going to unpack.
All right, so let's get into it, because to really get a handle on the errors, you've got to understand the blueprint first.
So what even is a chromosome, really? Okay, picture this. Take all of your genetic info, every single instruction, and imagine it's wound up super tightly into these incredibly efficient little packages. We get 23 of these from each parent, which gives us those 23 pairs in total. And this right here, this is what that complete set of 46 looks like. Now most of these, 22 pairs to be precise, are what we call autosomes. They handle pretty much all of our body's traits. But it's that last pair, the 23rd one, that gets a lot of the spotlight. The sex chromosomes. They're the ones that determine biological sex. This whole layout, that's our baseline, the standard human karyotype.
But like we've been saying, that copying process, well, it isn't always foolproof. Let's take a look at what actually happens when those copies get messed up during cell division.
Okay, so this is the moment where things can go wrong. See, when a cell splits to make new sperm or egg cells, those chromosome pairs are supposed to separate nice and clean. But, sometimes they just get stuck. This failure to separate, or segregate as it's called, is really where the trouble starts.
And this process, it has a name. Aneuploidy. You can see it laid out pretty clearly here. So you see the cell, it's prepping to divide. Then, bam! Step 2, a pair of chromatids, which are basically just identical copies of a chromosome that are still attached, they fail to pull apart correctly. And the result, as you can see in step 3, is that the new cells wind up with either one too many or one too few chromosomes.
So this basically leads to two main outcomes. You've got trisomy, which is when a cell gets an extra chromosome, so it has 3 copies instead of the usual 2. And then you have monosomy, which is the exact opposite. A cell loses a chromosome, leaving it with just one lonely copy.
All right, let's make this real. We're going to look at probably the most well-known example of a trisomy, Down syndrome.
And the cause is right there. It's a trisomy of chromosome 21. What that means is that a person with Down syndrome has 3 copies of this particular chromosome in their cells, not the usual 2. This gives you a really good snapshot. It was first described way back in 1866 by Langdon Down, and it's associated with some very distinct physical traits, like short stature, a small round head, and that single deep crease you often see across the palm. That extra chromosome also leads to retarded physical, psychomotor, and mental development.
Now, it's not just the autosomes that can be affected. This whole aneuploidy thing can also happen with that 23rd pair. Yeah, the sex chromosomes.
So let's start with Klinefelter syndrome. This is another case of trisomy, but this time it's all about an extra copy of the X chromosome.
So instead of the typical XY karyotype for a male, someone with Klinefelter's has an XXY karyotype. That means they have 47 chromosomes in total. And while they have what you'd call overall masculine development, that extra X chromosome can lead to some feminine traits like gynecomastia, which is the development of breast tissue. And a really key point here is that these individuals are sterile.
Okay, now for the flip side of the coin, Turner syndrome. This is a classic example of monosomy. So we're not talking about gaining a chromosome here, we're talking about the absence of one of the X chromosomes in a female.
The karyotype for this is 45X0. That means there are only 45 chromosomes total. And that absence, it has some really significant effects. These individuals are female, but because their ovaries are rudimentary, they are sterile, and they also lack other secondary sexual characteristics.
This chart really just boils it down, doesn't it? It's really all about addition versus subtraction. Klinefelter's, that's from gaining an extra X chromosome. Turner's, that's from losing one. Just 2 different kinds of errors, but they lead to 2 totally different outcomes.
So, okay, we've looked at these specific examples. What's the bigger story here? What's the big takeaway about our genetic blueprint?
I think the most important thing to remember is just how incredibly precise our genetic blueprint is. The gain or loss of just one single chromosome, one tiny little package of DNA out of 46, can have these massive, wide-ranging consequences for a person. It really just goes to show you how delicate that genetic balance truly is.
And that kind of leaves us with one final big thought to chew on. When you think about it, these chromosomal disorders are really just variations on the standard human blueprint. And if that blueprint defines so much of who we are, what are these variations, these typos in the manual? What do they really tell us about the incredible, mind-boggling diversity and complexity of just being human?